Hello, everyone. Welcome to Discover Brigham. Our first presenter is going to be Adriana Panayi from the Department of Surgery. Let's give her a warm welcome. And we just like to give you some logistics for the day. Each presenter will have 9 minutes to discuss their winning poster. There'll be 4 minutes for question and answer afterwards. So you can put those in the chat or raise your hand and we can unmute you. So without any further ado, Adriana, take it away.
Hi, everyone. So I'm Adriana. I work in the Division of Plastic Surgery, Department of Surgery, and I'll be discussing the project that we performed in Dr. Orgol's lab, the efficacy of autologous micrografts in a collagen-Glycosaminoglycan scaffold for skin wound healing. Just some disclosures. This project was funded by Stepping Strong and Integra, and Dr. Orgol is a consultant for Integra Life Sciences. In medicine, the ability to regenerate skin would revolutionize the field. Currently, the standard is using split thickness grafts, but the disadvantage of this, although they provide stem cells and allow for healing, the disadvantage of this is that they can result in scarring in the donor and recipient site, as well as they depend on an availability of tissue for grafting, and this is not always the case, especially in extensive burns. Currently, other techniques that are available are micrografting, so taking very, very small pieces of skin and grafting them directly onto the wound, as well as the utilization of scaffolds, which eventually biodegrade and offer promising results. A combination of the two has yet to be studied, and that was our goal, to combine the two techniques. And this may be promising for wound repair. The scaffold that we used briefly is a dermal regeneration template. It is a matrix of cross-linked type 1 collagen and glycosaminoglycan. It is porous, and it is FDA approved and has been extensively used in skin wounds and tendon repair. We utilized diabetic mice, so that's the DBDB mice, and the reason we used diabetic mice is because their wound healing is a little bit slower than wild type mice. And we split this mice into 4 groups, our 2 main groups, which were mice that received a dorsal wound, a full thickness dorsal wound, and mice which received a modified scaffold. We created the scaffold by taking micrografts using this pen tool. We took the micrografts and we embedded them in the scaffold, like shown in these pictures, and we grafted them onto the mice. We had further 2 control groups. One group received a non-modified scaffold, so just the square of a scaffold, and one group received the micrografts modified scaffold, but the micrografts were not defatted. So in addition to the epidermis and dermis, there was also a fat layer. We photographed the mice, and at specific time points, and we monitored the wound healing rate, as well as the viability of the scaffold and the micrografts. And as you can see, up to day 14 post-surgery, you could see the micrografts in the scaffold. There were no signs of necrosis or of infection. And in addition, the mice that were treated with the modified scaffold were healing at a slightly faster rate than the untreated group. We took skin tissue for histological analysis, and we performed 4 types of staining. We looked at hematoxylin, eosin to look at the wound bed thickness, Mason's trichrome to look at collagen deposition, Ki67 to look for cellular proliferation, and CD45 for leukocyte infiltration. We saw a significant difference in terms of wound bed thickness and collagen deposition. Both of these are factors that are used to assess for good wound healing. And in addition, we saw that there was no difference in the leukocyte infiltration, which is a good thing because that means there was no, the micrografts were not detected as foreign and there was no anti-inflammatory reaction occurring. We also performed some specialized analysis. We took some panel scans of the entire wound. You can see it here, and you can see the grafts inside the scaffold. And we assessed cell migration, cellular proliferation, and keratinocyte migration. And we did this by looking at 5 specific locations in the panel scan. The scaffold bed interface, the scaffold graft interface, the scaffold, which was a point further away from the interfaces, the graft, and the scaffold wound border interface. And we assessed the cellular proliferation and keratinocyte migration. In terms of cellular proliferation, we noticed that there was a difference, a significant difference in the wound bed thickness, the wound bed border and scaffold interface in comparison to the scaffold furthest away from the interface. This was also evident in the keratinocyte migration. So with keratinocyte migration being seen at the wound bed, at the wound border interface, as well as at the graft interface with the scaffold. Finally, we assessed the vascularization. In wound healing, good vascularization can be seen with an appropriate number of blood vessels, with the vessels being ideally mature. So we assessed the density and maturity, and we found that the group that was treated with the micrograft modified scaffold showed the highest density and maturity. And this maturity was significantly different to the group treated with the unmodified scaffold. So the grafts were themselves improving the maturation of blood vessels. We also performed some corrosion casting of the scaffold, and you can see the blood vessels in the scaffold. Finally, we propose a mechanism of how this how the micrografts improved wound healing. So in a way, the micrografts acted just as the wound border acts by allowing keratinocyte migration from their edges. They were able to, we hypothesized that they're able to release growth factors, which helped with cellular proliferation in the area. And they moved from the wound bed, from the wound borders into the center of the scaffold. To conclude, we saw an improvement in wound healing, an increase in cellular proliferation, collagen deposition, keratinocyte migration, and vascular maturation, all of which are indications of better wound healing. And in our future experiments, we would like to investigate this in larger animal models, such as rats and pigs, and as well as perform a human study, which, given that the scaffold is already FDA approved, would be an easy progression into utilization in humans. So overall, this experiment provides promising potential for clinical treatment of human wounds. Just some acknowledgements. I would like to acknowledge my team, some of which are present I can see are present during this presentation, and I couldn't have done this work without them, and my supervisor, who has been extremely helpful with this project. If you have any questions, please let me know now, or also feel free to email me. My email is on this slide. Thank you.
Thank you so much, Adriana. Are there any questions right now for Adriana that she could answer? If you want to, raise your hand or put something in the chat, we're happy to talk now. If not, can follow up afterwards.
Yeah, there's nothing in the chat right now.
Okay.
But if anyone thinks of anything, we can also take some questions for all 3 of the presenters at the end as well.
Right, then we will move on to poster winner, our second poster winner, Andrew Schaumberg. He is from the Department of Pathology, and there'll be about 10 minutes for the presentation and about 4 minutes afterwards for questions. please raise your hand or put anything in the chat if you have any questions. And Andrew, I'll leave it to you.
All right, thanks for that introduction. I'll share my screen. All right, are we able to see my screen?
Yes, we can see it.
Okay, so hi, I'm Andrew Schaumberg. If you take a screenshot of this, you can use your cell phone to check out our QR code on the left side here. That'll take you to our website. You can hear more about our poster and the talk, which is about PathoBot, twitter.com/PathoBot. It's a little bit like Google image search, but it's pathology specific. So specifically, it's an artificial intelligence driven pathology case search service on Twitter. So it searches patient cases both on Twitter and in the published literature in PubMed. We've published our method in Modern Pathology, which is available at this nature.com URL that you see on the screen. But what do these patient cases look like, you might ask? Well, here's our collaborator, Dr. Fularame in Lagos, Nigeria, and he's sharing a case on Twitter. So let's look at that. So the clinical impression of this is it's either two things, one of two things, either disseminated varicella for this patient or drug-induced toxic epidermal necrolysis. So we see 4 hematoxylin stained photomicrographs here. This is from excised tissue, and we see, some immune cells here. We see some dirty necrosis here. This patient is actually gravely ill. I'll show you that on the next slide. It's graphic, so if you're sensitive, look away now. So here we go. This patient is quite sick, a 20-year-old, 3-day history of generalized blisters that erupt. Also patient history of a relative with chicken pox. So my read on this is that this is a young mother in Nigeria. She's taking care of her child, and she herself is becoming gravely sick. So the question is, clinically, is she sick because of disseminated varicella infection or disseminated chicken pox? Or is she sick because she took amoxicillin the day before and she's having this toxic epidermal necrolysis as a result? So Dr. Fularame in Nigeria is asking this on social media. So Dr. Gardner, another one of our collaborators on social media, also a pathologist, chimes in that in his opinion, this is disseminated varicella. And he also mentions looking for a Nikolsky sign. Now, in Nigeria, there's not a lot of money to do specialized workups or order specialized stains, but Nikolsky sign is something you can do. So if you imagine this is a pencil here and you twist that against the patient's skin, if the dermis is thoroughly broken down with toxic epidermal necrolysis, Nikolsky sign, that twist will break the patient's skin. That skin won't break for disseminated varicella. And I believe the final diagnosis here was disseminated varicella, and the patient was negative for Nikolsky signs. So these informal opinions on pathology worldwide is a very real thing. It makes a difference. Indeed, Nix et al. Have found that pathologists in developing countries, such as Nigeria, often use social media, and 22% of the posts they analyzed involved asking for opinions on a diagnosis. So increasing access to physicians is actually crucially important. It's so important that the United Nations has developed their Sustainable Development Goals to make the world a better place for everyone. And number 3 on that list is health, good health and well-being. Increased access to physicians, of course, is an important part of that. If you're sick, like that gravely ill patient, it's good for you if you can see a doctor right away. And it's also good for you if your clinicians responsible for your care can get second opinions from each other. So we want to use AI to bring together pathologists to discuss the next steps in any patient's care and build consensus worldwide. Dr. Ho is one of our collaborators in Belgium, and in his case, we see some of the clues, a variety of clues that pathologists will include on these posts in social media, like hashtag breast path, it's breast related, hashtag GI path, gastrointestinal related, metastatic globular carcinoma in the ileum. This is a clue that there's potentially malignant disease in this case. We do a variety of tests, a variety of things with our artificial intelligence, the most important of which is this 3-category disease state task that we do. So the universe of disease could be considered as one of 3 categories. So is it non-neoplastic, non-tumor like this herpes simplex virus infection? Is it benign like this hamartoma here, any number of cysts? Or is it full-blown malignancy like this gastric adenocarcinoma here, sarcomas, leukemia, lymphoma, all fall into that category? So you might ask, how does this all actually work at a high level? Well, we start with recruitment in step A here. We recruit pathologists worldwide. If they consent to share the data with us, then we download their data, annotate that data, train an AI on that data to make a disease state prediction. And so then when a pathologist searches via our bot on social media, the bot can use the trained artificial intelligence to do a variety of tasks, like search social media for similar patient cases, search PubMed for similar patient cases, and also post predictions all on Twitter about the evidence of disease predicted for that case. Is it non-tumor, benign, low-grade, or full-blown malignancy? So pathologists can interact with us, interact with our tools, see the bot in action, and sometimes they're inspired to collaborate with us. So we've effectively looped back and done recruitment again. So we can download the data, annotate that new data, train our AI again on the updated data. It's more data, so we do better. And of course, then the search is better because we've expanded our database of searchable cases. So how does it work in practice? Well, Dr. Stangerman in Honduras is saying, dear #PulmPath, pulmonary pathology, this is lung. Here's a female in her 70s, there are multiple pleural lesions evident. What's your favorite diagnosis? And would you order any special stains in your workup of this patient's diagnosis? She mentions PathoBot by name explicitly in her post to Twitter. So PathoBot responds in real time with the top 8 most similar patient cases in our database. These are all links. So this links to other pathologists' cases. This is the most important thing of what we do. We link to similar pathologist cases. That linking notifies other pathologists. So they're in effect invited to comment on this case. We want to bring pathologists together to build consensus and discuss the next steps in any patient's care. So that's what we do. PathoBot also responds to itself after searching PubMed Central for published case reports in the literature for the 8 most similar patient cases there. PathoBot responds to itself again with a per image breakdown of the predictions for disease state for each image, of course. So is it non-tumor, benign, or malignant? Here, malignant is highest, so this is evidence predicted that this is malignancy here. All those predictions, all those probabilities are synthesized together for an overall prediction for the patient. This is an important sanity check. Prediction is a sanity check for search. If prediction is wrong, if it's saying non-tumor, but it's obviously a malignant disease, if it's wrong, then a pathologist knows to discard the search results too. It's all the same AI, so sanity checks and interpretability are really important for any medical facing application, even if it's research only purposes like ours. So how does it work at a very low level? We use deep learning, artificial neural networks, deep neural networks to make these predictions. We train it on our pathology data from social media. We include these tissue type covariates and other covariates that describe the tissue, like pulmonary or other tissues, to include all that in an overall prediction for the patient. And we include some hand-engineered features as well for describing color and edges and texture in a human algorithmically defined manner. And we use some machine learning on top of all of that to give us some interpretability. So we can interpret this method at a very deep level. We can see what the deep neural network represents before training and after training on our data. It's very important to open the black box of AI. So we notice before training, color is important to add, tissue information is really important to add, but after learning, the deep learning has learned to represent those things. These are less important now, but texture becomes important. So it's all the more reason to suspect that blurred images that don't have visual texture information may be especially challenging for this AI. It's important to know that in advance, what it's doing and what it's not doing. We also get localization for free. So no pathologist ever circled where the hyperplasia was in this image, but in the center, we see high activation for low grade or benign disease. So these hyperplastic cells are evidence of that. And around the periphery, we see activation in yellow for non-tumor or normal tissue. So we get localization for free. So in conclusion, our project has become a globally distributed expert system that facilitates pathological diagnosis and brings expertise to where it's needed in hospitals and underserved regions. This is the first pan-tissue, pan-disease method for prediction and search on social media, and the first pathology study tested prospectively in public. We expect our project to cultivate a more connected world of physicians and improve patient care worldwide. But wait, that was all published. What's actually new? What are we bringing to this presentation? It's attention. So attention is really important to focus on what's important, right? In a photomicrograph. This is a photomicrograph. There's a circle here. A microscope has a circular field of view. Everything around that is just black, doesn't make any difference. So if you split that into a 5x5 grid, you'll see red hot attention right down the barrel of that microscope, so to speak, and ice cold blue attention around that. So it's effectively cropping that out. There's no diagnostic information in the black regions, or at least there's less. Pathologists also annotate their images. So this is a signature in Mandarin from our collaborator Hongyu Yang here in the United States. And you'll see ice cold blue attention around his signature. There's red attention or red hot in the pathology everywhere else. And that's potentially diagnostic. So we think this attention mechanism allows us to make more precise predictions and search potentially in the future. There's more work to do with that. So this is our poster. It talks more about the attention. You're welcome to screenshot this, check out that QR code with your cell phone. It'll take you to our website for more information. Attention pathologists, please contact me at this email address. If you'd like to collaborate and for postdoc engineering or research assistant positions, please contact my PI, Faisal Mahmood here at the Brigham. And thanks very much for your attention.
Thank you so much, Andrew. That was an exciting presentation and very cutting edge. Does anyone have any questions for Andrew? If you do, you can make a note, either type the question out in the chat, or you can just make a note in the chat that you have a question and we can unmute you if you'd prefer to ask it live. Okay, I don't see any.
Shy today.
Yeah, I don't see any questions coming in. But again, if you have some question at the end for either all the presenters.
There is a question.
There's a question.
Hey, all right.
Okay. Okay, Rohan.
Maybe I'll stop my screen sharing and talk.
Okay, I'm gonna unmute Rohan.
Okay.
Rohan, can you, is your microphone working now?
Or if you'd like to type, that's fine. I'll talk at you.
Hi, hi Andrew. Can you hear me now?
Yes.
Yes.
Hey, amazing work. I really like the work, and it's really interesting that how you use the social media, the images there. But then I just wanted to ask. I'm pretty sure you probably have acknowledged this already, but how do you plan on continuously making your model better or the algorithm better? So suppose if someone has uploaded an image and your model has misclassified it as something else, you obviously do say that okay, this is the confidence of the prediction, and you obviously give the metrics, but then how do you plan to use that data to probably make your model better so that it does not misclassify the next time?
Right. Well, that's a great question. So how do we learn from updates and mistakes, and how do we do better? There's actually a variety of answers to that. So if we're wrong or systematically wrong, we could revisit our data and see if any annotations are wrong. We could also pull more data from our collaborators to potentially do better for cases like this, or ask for that entity to be more represented in the cases some pathologists share. There's also, in the live PathoBot tool, there's a text component, and the text component is actually an important part of search. So for the paper, we focus just on the images. So without any text clues, how does search perform? And that's easy to measure, because if you measure accuracy in terms of, are you finding the same disease in your search hits relative to your search query? If those are the same, you're more accurate and you're doing better. But sometimes the text processing can be improved as well. So sometimes we don't understand on the text algorithm side what the pathologist is saying, or we're not inferring correctly from the whole scope of discussion in a Twitter thread, and we try to use as much information as possible. So sometimes it's an issue on the visual side, sometimes it's an issue on the text analysis side, but there are a lot of knobs we can turn to try to do better and improve quickly from any mistakes that we make. So it's a really important question, I think, that you're asking.
Right. Yeah. So I was just wondering if we can continuously use instance learning just to improve the model or something like that, so that it can just take care of the outliers or something. Yeah. So yeah, amazing work. I really loved your work, and
it's true. Yeah, and that's a nice use case of neural networks because it's online learning. It would be nice if it were a little bit smoother to pull data updates all the time and continuously
Right.
update the model. Unfortunately,
Something like federated learning would like be a very good application here in this, like continuously improving the model from the different kinds of like data all around the world, and yeah, pretty interesting.
it would. There's a tricky part too, where if you have a pathologist that's searching, they may or may not have consented to share data with us. And right away, like immediately, they may be sharing vacation photos or something that's not necessarily pathology. So that's why we always go through a curation process to make sure, everything's okay. Because sometimes, if you're very rude to a doctor, sometimes they'll take a picture of your car that includes a license plate and they'll say nice things about you on social media. So that's not training data for us. That's a different task.
Yeah. Thank you so much. Thank you so much.
Thank you for the question. That's a good question.
Okay.
Thanks again, Andrew, for your presentation. We'll now move to our third poster winner and our last speaker today, Anjali Sivakumar, who is from the Department of Medicine. And I will leave it to you, Anjali, to start your presentation.
Thank you, Lisa. Good morning, everyone. I am Anjali Devi Sivakumar. Today, I'm going to present our work on COVID-19 diagnosis, which we have been working at Dr. Shafi's lab. So, you all know that COVID-19 has been the biggest threat around the world since the start of this year. It has infected 51 million people around the world, and it has also killed like 1 million people around the world. And during this situation, the early diagnosis is really essential in order to reduce the spread of this disease. So you might have heard that a lot of tests has been introduced around the world, and the most widely used test is the PCR-based test. But the PCR-based test is really complex as it involves like a lot of steps like nucleic acid extraction, amplification, and in turn it requires like complicated equipments to perform this procedure. So we need to, only the experts can perform this procedure and also in a laboratory setup. So, and this test, even though it's really accurate, it's also time consuming. And because of the complexity of this test, we can perform it only in the lab. It's not accessible to people living in remote areas. On the other hand, if you look at the other test, which has been used widely, it's the paper-based antigen test. Even though this antigen test might be widely used, it's not accurate as the PCR-based test. So with so many problems in our current COVID diagnosis methods, you would wonder, why don't we have a simple diagnostic method that generates an easily detectable signal instead of a complex one? So we had developed two methods, both the molecular-based diagnostics and an antigen-based test, which actually generates bubbles on detecting SARS-CoV antigens in nasal swab samples. So in this presentation, I'm only going to talk about our antigen-based test. So in order to perform a diagnostic assay, we had developed a microfluidic chip as shown here. So the microfluidic channel of this chip is coated with antibodies specific to SARS-CoV-2 antigens. So once you load the sample into this chip, the virus actually gets attached to these antibodies. So we have captured the virus inside the chip. So now we are going to label the captured virus using platinum nanoprobe. Now we know that virus is actually present in the chip, but we want to visualize this using a smartphone camera without any additional optical attachment. For this purpose, so we catalyze the platinum nanoprobes, which is attached to this virus using hydrogen peroxide, which generates bubble in the microfluidic channel. So if you see the two microfluidic chip images here, you can see that bubbles are actually generated only in the presence of SARS-CoV-2. So in order to give you an idea of how actually the bubbles are generated, I have a time-lapse video of the bubbles being generated in the microfluidic channel. So you can see that the bubbles are progressively being generated in the channel. So moving on, we wanted to know whether our diagnostic assay is only specific to SARS-CoV-2. So we had tested a diagnostic assay with different viruses. So if you see the microfluidic chip images, which is on the top, you see that the bubbles are generated only in the microfluidic chip tested with SARS-CoV-2. So this shows how specific our assay is in detecting SARS-CoV-2 antigens. So on the other hand, if you see the microfluidic chip images in the bottom, you can see some difference in the configuration and also the morphology of the bubbles generated in the microfluidic chip. So initially we used to count these bubbles manually to establish a relationship between the concentration of the virus present in the sample. But later we found that instead of manually doing this, why can't we feed these images into a machine learning algorithm, which actually does the classification task for us? But usually machine learning algorithm requires a lot of data in order to give you accurate results. But during the start of a pandemic, we'll not have access to a lot of samples. So initially we had access only to 35 patient samples. So we got 35 microfluidic chip images. But this data is not sufficient to train a machine learning model. So what did we do? We had actually used a similar assay for testing HIV, HPV, and other viruses previously. And we had collected around 700 microfluidic chip images, but this image was not sufficient, still not sufficient for the machine learning algorithm. So if you see it in this figure, these are actually data points of a single disease. So each point here represents a microfluidic chip image. So this is actually an image of a microfluidic chip. We have actually cropped the channel and sequenced it in such a way that it can be processed by a machine learning algorithm. As I was talking to you earlier about the insufficient, like we don't have enough data for the machine learning model, like what did we do to, now I'll tell you how we overcame this problem. So actually there is an algorithm called generative adversarial network, which actually has the capability to generate more images which look similar to a real image. So we use this algorithm to generate around 17,000 fake images from the real images, but these we were not able to we can't distinguish. If you see the image generated by the generative adversarial network and the real image, you'll not be able to distinguish which one is real and fake. So with the help of this algorithm, we were able to generate 16,000 images. And if you see the microfluidic chip images at the bottom, these are the images actually generated by our adversarial network. So now in total, we had like around 17,000 images for a machine learning algorithm, which is more than sufficient for training the model accurately. So in order to classify the microfluidic chip images, we had used adversarial neural networks, which actually first learned the essential features in our microfluidic channel and then later classified them into positive and negative classes. So now that we have developed a proof of concept for our diagnostic assay, first in generating the signal and then later recording it and also processing it into useful data, we had tested this whole diagnostic method with 62 different nasal swab samples. So we had cross-checked this nasal swab samples with PCR to find out the ground truth whether they are positive and negative. So we tested these nasal swab samples with our diagnostic method, and from our results, we could see that our algorithm actually performed really well in classifying them into positive and negative with a limit of detection of 1,000 copies per ml. So if you look at the current antigen-based test available, the limit of detection is around 1 million copies per ml. But we were able to develop an antigen-based method with limit of detection of 1,000 copies per ml and also with a good accuracy. And by now we would have understood that our method is both simple, accurate, and also it can be easily adapted to a new emerging pathogen like SARS-CoV-2 with a limited data. So now that we're able to establish the concept of a diagnostic assay that can be performed in the lab, we had a thought like, why can't we integrate the whole thing into an automated system like here, so that like once we insert the sample, patient inserts the sample inside the device and just presses a button, the test would be conducted. So in order to develop our automated diagnostic system, we had actually used a cart. There are two components in our diagnostic system. One is the cartridge here, where we load all the reagents required for performing the diagnostic assay serially, so that it serially loads them into the microfluidic chip, which is inserted here in the cartridge, and then performs the diagnostic assay. So in order to like automate this process, we had used a microcontroller which times these loading of the reagents into the microfluidic chip. So I wanted to give you an idea of how our system actually works. So this is how it works. It is inside our device. So first we capture the virus. Later we wash out the unreacted virus from the microfluidic chip using PBS. Now the virus is actually inside this microfluidic chip. like as I was talking to you earlier, I told you that we are going to label these captured virus using platinum nanoprobe. And we are going to wait for some time so that all the viruses get labeled properly. Then we are going to wash out the unreacted platinum nanoprobe using PBS. Then later, now that we have both labeled and captured this virus inside the chip, we are going to catalyze the platinum nanoprobe using hydrogen peroxide. So after it catalyzes, and after some time, the bubbles would be generated. So this is actually a time-lapse video with color liquids in order to give you guys an idea about how our system actually works. So we are hoping that our diagnostic assay with our automated system could actually serve the needs of any upcoming pathogens. So firstly, I'd like to thank my mentor, Dr. Hadi Shakti, for giving me this opportunity to present this project and also work on this project. And I'd also thank the whole team who had worked really hard during this pandemic to make this project a success. And thank you so much.
Thank you, Anjali, for that really interesting presentation. Does anyone have any questions, either in the chat, or we can unmute you, raise your hand? Or if not, I think we have everyone's contact information in the program, so you can probably reach out to any of the speakers with any questions that come up afterwards. And with that, I suppose we will conclude our poster presentation session today. Congratulations to everyone who won. They're really interesting, great posters. I feel like I learned a lot, so thank you.